Dr. Marguerite Butler is with us today. Dr. Butler is an associate professor in my department in biology. She received her bachelor's and master's degree at Rensselaer Polytech Institute in New York and her PhD at Washington University in St. Louis, Missouri. She's interested in how animals function, and the interaction between morphological design, selective pressures, and evolutionary history. She's especially interested in sexual dimorphism, where males and females differ in ways that are important for their function or ecology. Current and recent research projects in her lab include locomotion in iguanas, sexual dimorphism and the cost of reproduction in lizards, and color vision, target detection, and behavior in Hawaiian damselflies, which she'll be sharing with us today. Help me welcome Dr. Marguerite Butler.
That's a really nice introduction. I should put that on my web page. Thanks. Hi, everybody. I see many of you that I know already, so that's awesome. Today I'm going to talk to you about some of our research, as opposed to a lot of the teaching that you folks know me through. So for, I don't know, about 15 or 20 years I've been working on damselflies in Hawaii. And they are really, really cool native insects. They're big. They're about like this big. So they're not flies like fruit flies, you know, or house flies. They're damselflies. They have very typical insect body parts: the head, a thorax where the flight muscles are, and then abdomen. But in Hawaii, we've gotten many, many different species, about 20 different endemic species. So what happened was, okay, can you, okay, sorry. So we have about 20 species of these native damselflies. So as you probably have heard over and over again, Hawaii is the most isolated archipelago in the world. So these are flying insects, but they don't fly very far, or, you know, so for one to get to Hawaii is a very, very rare event. But once they arrived here, they're able to speciate. And so we have over 20 forms, and many of them are ecologically distinct. This guy, this really cute, charming guy, is at Lion Arboretum. Okay, this is Megalagrian nigrolineatum, and it was just recently listed on the Federal Endangered Species list. Personally, I don't think it's endangered, but it's on the endangered species list. It's actually the most common damselfly, native Hawaiian damselfly on Oahu, but that's okay. Okay, anyway, they have enormous eyes, as you can see. This is its closely related sister species, Nigrohomatum, and this one is found on Maui. So they're really beautiful. They're very colorful, and for the most part, oftentimes every volcanic range has a different species. So there's all kinds of things going on that could make you think about sexual selection leading to speciation or things like that. So it's an adaptive radiation, and this is their distribution across the Hawaiian Islands. And it's thought that they arrived about 12 million years ago, which of course is older than Kauai, but as you know, there were islands before. So the thought is that there has been both in-island speciation as well as a rare migratory event, which then leads to other species forming. So overall, you see this progression of numbers of species. So the oldest island has the most endemics, and the youngest have fewer endemics. Okay. So what's really interesting about Megalagrian is that they use these really different breeding habitats. And so so what we're thinking is that this genus sort of evolved along with the evolution of the islands. So, you know, as you know, we live on volcanoes. So we have a volcanic island forming, and what happens? We get erosion, right? So we have rainfall. So with rivers and streams forming, we have erosion, which creates all these valleys on the different islands. And then with the process of erosion, you have different habitats forming, so that, you know, perhaps the older islands have more niches or earlier habitat availability than others. But anyway, okay, here's the different habitats that they use. They use big open streams, and they tend to be big species. They also use the pools that are on the sides of streams, which have like slow moving water, and sometimes they're in the deep shade. They also use seeps. So this is really weird. Seeps are like mini waterfalls. So these damselflies lay so all damselflies are basically aquatic insects. They lay their eggs in the water generally, they hatch into larvae, and they go through all these molts, and then they emerge into adults out of the stream. But some of them don't lay their eggs in the stream or near the stream. They lay them on the walls of the valleys that are mossy, that are wet. And then the weirdest ones of all are the terrestrial ones. So we have some that have actually evolved, and they lay their naiads either in plants or in the underneath these really scraggy ferns. And what's really amazing about Hawaii's damselflies is that this small genus of 20 species spans this incredible ecological diversity. And if you were to look worldwide at damselflies, you would have to consider the entire world to find these 6 ecological types. Okay, so it's incredible ecological divergence. Okay, so this is their phylogeny, or their family tree of the different species. And so the colors indicate the ecological types. So we had a divergence between the pool and the stream species, and then the seep species are in green. Okay, and I'll get back to that later. So these are some of the habitats. So the stream, like I said, is usually really wide open. This is on Kauai in the I'm having a mid-age brain fart. Sorry, early Alzheimer's. Kokee. It's in Kokee. Okay. Anyway, big open stream, fast-moving water, really sunny. And so the stream damselflies tend to be pretty big and strong flyers. This is what a pool habitat looks like, and a pool habitat can be an isolated pool or a very small stream or a tributary. And here's where you'll find, here's where you'll find typical aquatic larvae, naiads. This is a seep, and it's in the bog in Kamako Preserve on Molokai. This is one of the weird plant species. So these eggs are laid in the axils of this Ie, or Astelia plants. And these are bromeliad-shaped plants, and so in the axils of the leaves, there might be like a thimble full of water, and that's where the eggs are laid. And so these guys are pretty, more or less terrestrial. They have thickened cuticle, and they can crawl out. They stay damp, but they can crawl out and be exposed on the regular leaf. And then the terrestrial ones, we actually have never found any as larvae, as naiads. We only find the adults. But back in the 30s, Williams, who was a famous Hawaiian naturalist, basically what he did was he cleared like 2 acres of uluhe fern and went through the leaf litter and found the naiads. Yeah, I'm not going to do that in 2016. But they're thought to live under this. And if you've been out in the field, this is really tough, dry stuff, very craggy, very hard to get into. Okay, so those are the habitat types. This is a seep, and this is Julio, one of my grad students, and he's going to tell us.
So we found a female Hawaiian seep, Megalagrion Hawaiian seep, flying, ovipositing along these roots that are attached to the seep. Basically, she would oviposit one egg, fly about 5-10 centimeters, oviposit another egg, fly up again. Saw her do that about 3 times, or I caught her.
Wow. So this is a seep. Okay, so they have moved out of the stream. Okay, and this is what one of the females looks like. This is a blue species. Okay, and this is what some of our fieldwork looks like. And we go to these streams and we, prepare for the day. We catch damselflies. We collect ecological data. And one thing to note is this really huge gradient of light, and it shifts throughout the day. So that had us thinking, what is it that's giving rise to this amazing diversity of species? And at this stream, at one time, you could find 6 different species there of megalagrian. So we were trying to figure out what could it be that would promote this kind of adaptive radiation. And so what we've been focusing on is related to their color. So a lot of different species are different colors, and a lot of males and females are different colors as well as their vision. So we're looking at differences in light levels, differences in ambient lighting, differences in body coloration, and their use of the habitat. Okay, so these are some of our tools, tabbies and nets. And this is Christina and Julio, who were super excited about catching damselflies. And this is another species. Okay. So, so animals use color for many different purposes. A lot of it has to do with social signaling, but it could also be camouflage, predator escape. A lot of it has to do with attracting mates or defending your territory against conspecifics. It's either you want to be seen or you want to hide. And in megalagrian, visual detection is important, like all damselflies. They also use it to find mates, they avoid predators, and also to find their dinner. So they do everything in flight. So they catch their prey in flight. They catch their mate in flight as well. But they do perch and sort of like observe what's going on around them. And of course they have these humongous eyes, which are almost like a 360-degree array around their head. So we have been looking pretty carefully at their eyes and how they're designed and whether it helps them, because as you saw in a lot of those pictures, they live in dark habitats, which is really weird. Most mainland damselflies live in these bright, sunny, open ponds. But they tend to pick dark places, well, a lot of them tend to pick dark places to live. So we're trying to figure out why and how can they actually do this. So insect eyes are made up of ommatidia, and you can see that better in this close-up. So each of those little things is the lens of an ommatidia. And inside an ommatidia, there's an array of opsins or receptors. There's 9 opsins within each one. And so in order to see, you have light coming in, focusing on the receptor and exciting the cell. And they're different from the design of our eyes because it's like a pixel array. So in order to see an image, you have to have some pixels light up and others not. So you can kind of think of it as a pretty grainy TV. So the more they have, the sharper the details of the image that they can resolve. But there's a trade-off because the more you have, the smaller each of them are. And so you're going to limit your ability to capture light. So there's a big trade-off between resolution and light capture, resolution and sensitivity. So because they're small insects, they can only have eyes that are so big. So we're trying to figure out how can these small insects live in a dark environment, see really well, and capture everything in flight. So that's what we've been focusing on. And so the 2 main design parameters are facet diameter, which relates to light capture, and interommatidial angle, which relates to their acuity or resolution. And so we measured this for a lot of, so here's just to show you. So you see the male coming in right here? They're really fast and they're very, very agile in flight. So the male's gonna come in right about here. You see? Yeah, they're really fast. And And then they also capture prey in flight as well. So here's the damselfly, and then a bug is going to fly by and it'll go right off after it. So yeah, sorry, it's really, really hard to capture actual predation events in these guys. Okay. So this is Julio out measuring light.
I am about to take some light data so we can study the visual adaptations of these damselflies.
Which damselflies?
This is Megalagrion blackburni, the largest damselfly in Hawaii.
And does he usually sit on your lap?
They're usually very skittish. This is actually amazing. We actually don't come close to them, ever.
Yeah, so we've been able to go to some amazing places, and this was a site on Molokai. But anyway, what we do is we go to where they live, we measure their light environment, we measure their eyes, and we try to see if their eyes are adapted to the light environment that they live in. Okay, so this was analysis of eye morphology. So we took photos of the eye and measured their facet diameters and the angles by which they're separated. So this gives us an idea of how much light sensitivity they have and how much resolution ability they have. And you can see that right here, you see this big black spot is indicative of a really high acuity area in the front. So we can model, so basically their best vision is right in the front. That's what we learned. And so we can figure out where they're pointing and figure out how well they can see in that direction and measure the light in that direction. Okay, so we're trying to understand this radiation and how they're adapting to the environment. And they have a constrained visual system. So basically, the visual performance is limited by the surface area of the eye. So you could potentially evolve to have increased light sensitivity. So you would get more light sensitivity by making your ommatidia bigger, fatter. Okay? You're gonna, of course, reduce resolution though. You could, alternatively, really optimize resolution, in which case you're going to make a greater density of receptors, which means that each one is gonna be smaller. Okay, sorry. Finally, another possibility is that they might have regionalization across their eye. So they could potentially have a big fat ommatidia right in the front, in the middle, and smaller ommatidia around the side. So that way you can sort of have a little bit of the best of both worlds, but still maintain a reasonably sized eye, given that you're so small. And so I'm not going to show you the data, but this is basically what we found. We found that Nigrolineatum, this guy, which is one of the smallest species, has the biggest facet diameters because they have this huge enlargement right in the front. So they can stay small, live in these tiny little dark habitats in little puddles of water, but still be able to function because they were able to make the front of the ommatidia in the front very large. We don't find that same trade-off in the big species. They seem to be okay, so they have more evenly sized ommatidia all the way around. So this was really cool. This is the first evidence we had of adaptation to the environment. Okay, so now I'm going to tell you a little bit about, and now we're working on all those light measurements that we took, and we actually are finding that different species do preferentially choose different light environments, and they seem to have adaptations that best fit them for those environments. So we're super excited. Sorry if that sounds geeky and boring, but we are super excited about that result. Okay, they are amazing at color vision. So if you look, so we have actually measured in a lab in Japan with a electrophysiologist, I went and I took these damselflies there and we wired them up. So what you do is you make these very fine capillary electrodes. You cut a little window through the lens of the eyes and you poke their eye. And so you can't be squeamish. But what you're doing is you're putting the receptor in a single cell, in a single ommatidia. And then you shine light on it of different wavelengths, and you measure the electrical excitation that you get back from it. So you can characterize the sensitivity of each cell in the eye if you so chose to actually map the entire eye. So here's what I learned. So we got the data, and then we fit these templates. Sorry, this is technical. We fit these different templates. So I was able to find out that they have at least four different receptor types. So they have a UV, a blue, a green, and a red. So they have really good color vision, way better than us. You know, we don't have a UV receptor. They may have more, but this is just what I could measure physiologically. Okay, so the really fun thing that you can do with these guys, because they're kind of big for insects, and they're pretty, sometimes feisty, is you can actually do experiments with them. So we have been doing these color cue experiments to figure out, okay, so they have these amazing eyes, they have great opsins, do they care about colors? Can they really see those colors? So if they can see those colors, and those colors are important to their life, they should have really good behavioral responses to them. And that's sort of the final cap, I guess, to the proof that they do see in color. So although the forest you think looks really, really green, especially on the scale of an insect, there's a lot of different colors there. Okay, so color could be really important because you have to be able to discriminate important cues from irrelevant cues. You don't want to be like chasing after everything, you'll waste all your energy. So we did these bead experiments where we hung beads on fine tungsten wire, and they tend to perch. So this is at Lion Arboretum. We went to where they are, we showed them the bead, and then saw what they did. And this was primarily the work of Christina Lincoln, who was an undergrad researcher in my lab, and she did a tremendous amount of work. And she made all kinds of really cool discoveries. So she figured out that so these are two different morphs of Megalagrian nigrolineatum, the green-eyed one and the blue-eyed one. So we knew that, but what we didn't know was that there's about like 14 different color morphs of males in these populations. And she discovered that the thoraxes differ amazingly. You know, why would you have all these different stripes? You know, so she was starting to think, oh, there might be different behavioral strategies. Some of these are maybe more territorial or more aggressive, and some are sneakers and things like that. So there's all kinds of things that we could potentially find out. But it does correlate with other things that are known about animals that live in dark habitats. They're often brightly colored. Anyway, so she did do these reflectance measurements, and different body parts are really different colors, and there's clear sexual dimorphism. So they live in a dark habitat, but they're very bright. They're sexually dichromatic. Males are polymorphic in the face and the eyes, as well as the thorax, and in fact, they're hyperpolymorphic. So it seems really crazy that they would have these really bright colors, but it makes, if you think about it, if you want to live in a dark habitat and you have to communicate, you do want a signal that you can broadcast. And so Christina's idea is that the eyes are the attention-getting signals. And they sort of indicate, hey, I'm a Megalagrian nigrolineatum. And then she thinks that the thorax color is variable and could be an indicator of quality, male quality or female quality. And they're really amazing because they live in such dark habitats, and you see how the eyes are red on top and really bright on the bottom. When the animal turns to face you, the eyes just bing, like even as a human, that you're so much huger when the animal turns to face you, you can actually see those two little dots in the dark habitat. They really are like little flashlights. But when they turn to the side, what you see is the red on the top of the eyes and the black, and they disappear. So it's pretty amazing. They can show themselves when they want and disappear when they want. Yeah, so this is the bead experiment, and so this is the presenting color. And then this was an example where a male attacked. And so she scored attack, track, avoid, and no response. And so attack was when they flew right up to it and touched it. So we found out that they're very aggressive towards black, also blue and green, but not red, which was very curious because red is kind of the male color. But they're most aggressive towards brown, which actually is limited to females. So we're learning some really interesting things about their behavior. And we've learned that they can make fine distinctions in color, and that different colors can provide different cues across species. We also did this experiment in Xanthomelis, which is a species that lives at, oddly enough, Tripler Army Medical Center. It was thought to be extinct from this island. This species exists on multiple islands. It was thought to be extirpated from this island altogether, and then when they did an environmental survey for Tripler, they discovered it there. It's living in a crappy little ditch. It's a drainage ditch that's fed by a pipe. So if somebody turns off the hose, the thing dries up. But it's getting listed also. So anyway, the Army has been great. The Army is a very good promoter of environment, and they maintain the stream for this species. So it's hilarious, but it's a great species and it's easy to study. So anyway, in this species, they were significantly more aggressive towards red, which is the color of males. And they tracked brown, which is the color of females. So in this species, it was very important for them, and it did seem to make sense with regard to how they responded to conspecific males and conspecific females. So it's even more curious when you consider that many of them look very similar. So these are 3 species of red damselflies that you can find in the same stream on Kauai. In fact, these are from the same stream on Kauai. And so, the question is, how do they tell each other apart? So we have also been measuring a lot of these different colors. Okay, I don't think I'm gonna go through all of this. Okay. So this is what a lot of the canopy looks like above a stream. And sometimes they're very, very dark. So this is just showing you how we measured the light. So we actually found where they're perching and measured the light in the different directions, straight up and also in the directions that they're pointed. We actually looked at the darkness of the habitat, and we found out that that the heterogeneity of the habitat is really important to them. So this is something that we found out by doing these careful measurements. Instead of just taking one measurement per habitat, which is common in vision studies, we actually measured everywhere that they perched and compared them to random locations in the stream. And then we looked at whether there was structure in the light, thinking that it's the stream itself that creates the structure. So the canopy should be thinnest above the stream, and then so we were asking the question, do they use behavior to exploit the light geometry? So they're in a limited environment, but can they basically extend their abilities by behaviorally exploiting bright spots and things like that in the environment? Okay, so we first found out that their habitat is really dark. Okay. But for species like Xanthomelis, although they live in a dark habitat, they can choose bright spots within the habitat, and they do so. So they, even though the overall habitat is dark, they can be selective and sit in a sunspot so that they're obvious to conspecifics. They also tend to perch at a slightly upward angle where they do choose to perch. And so if you measure the direction that they're perching, their forward direction sees a brighter background than other directions. So even within the spot that they're perching, they'll turn in a way that's advantageous for them. So does it matter? We tried to figure out if it mattered by modeling what this means for their ability to detect conspecifics. So we looked at contrast, and we calculated, okay, so this is a color space map, and so we looked at the amount of contrast that they get, and so this envelope are all the samples of the background habitat. And the mosquitoes, so we also measured the reflectance of mosquitoes, and mosquitoes blend in really well with the habitat. So the way that the direction that they're looking doesn't help them to see mosquitoes any better. But it does help them see females a little bit better, and it helps them see males a lot better. So these little differences in how they exploit their environment really do matter. Okay, so yeah, we were really excited about this because this is to our knowledge, the first demonstration of an animal actually selectively pointing in a direction to look at backgrounds that make conspecifics more conspicuous. So they're competing males or females, but not food. So, okay, so the big lesson is sex before food. Right? That's what's important. Okay. Okay, so anyway, so what's the whole overall story here? These damselflies actually have, so other animals have more extreme visual adaptations. As you know, there are insects that fly at night, right? There's nocturnal insects, but they have really different eyes. They have what's called superposition eyes, which involve neural rewiring and stuff. These guys have the eye morphology of a typical day-active insect. There's no apparent specialization for dim light. And the habitat is very dark, and light is limiting. But what we found is that they do have some small adaptations, like sometimes they make their omentity a little bit bigger. And more importantly, they use behavior to extend their visual capabilities. And this is really fine-tuned. So this might be how they're able to exploit all these different niches that Hawaii has to offer. And also, it's both the signaler and the receiver that play a critical role in making this all work. And we also found out that it's important to measure how animals actually use their environment, not just some sort of global average. But you have to measure the heterogeneity of the light so that you can actually see any interesting differences in the scale that they really use their habitats. So here's what I showed you at the beginning. And so how does this help us understand their adaptive radiation? Well, maybe about 12 million years ago, the first megalagrian arrived, probably on a different island before Kauai. And then what you have here is an early divergence. And you get the evolution of the stream species and the pool species. Okay, so this makes sense, and it's very generic as far as damselflies habitats go. Most damselflies use streams or pools. And then you have diversification going on. And then at some point when Kauai arises, you get the evolution of many more species. So you're getting a lot of diversification of pool species and seep species. These boxes are what island they're on. So the black is kauai. So you can see that there's a lot of kauai boxes. And then red is Oahu. And then yellow is Big Island and blue is Maui. So you had the evolution of these different habitat types arise at about the time that Kauai was available. And so this is really a story of ecological opportunity giving rise to more species that are occupying these different niches. And then you have speciation going on to different islands. Oh, sorry. I didn't mean to include that. Okay. So that was the serious part. Now I'm going to just show you some fun slides and stuff. So this is Heterogamius from Molokai. This was just the weirdest place. Molokai, everything is slow and everyone is friendly, including the damselflies. You could practically just pick them up. It was really crazy. But there is all kinds of mating activity going on, and they're right next to the stream where they're laying their naiads, their eggs. Okay. This is what some eggs look like. Many of the species lay eggs in plant stems. So they don't just shoot them into the stream, but they do typically lay them on vegetation or something like that. And this is a naiad right here. And this was crazy. So this was on Maui. We caught some naiads. Oh, good buddy.
Okay, I'm probably
Oh my god.
Oh my god. He's a great crawler.
Isn't he? We just pulled him out of the stream.
Wow.
We were shocked they could walk.
Actually, amazed there. They was like.
I didn't know they
really
could crawl like that.
like that.
He just fell over.
Okay.
He's trying to prove us wrong.
Tell me, Christina, what you're doing.
Okay, so I just caught this naiad from the stream. This is a megalagrion naiad, so it's a native Hawaiian. It's going to be a native Hawaiian damselfly someday. We're not sure which species, but it's maybe going to be blackburni, califia, or Hawaiian sea.
Wow. And do they normally crawl like that in your hand?
I have never seen them crawl. They're known to be really good swimmers and good to be in the water, but we didn't know they were so good terrestrially.
Wow. So where'd you find it?
I found him on this rock right here.
Wow.
And he just swam right in my hand.
That's awesome. That's such long legs. Yeah, that is weird that they have such long legs. Okay. So we caught a bunch of naiads and we brought them back to the lab and we did a float tank experiment. So we were curious about their stream adaptations and whether they're adapted to hang on to swift moving water. And we were really lucky. We were able to compare damselfly naiads with dragonfly naiads. And so originally I started studying iguanas as Cindy said. So I'm a vertebrate biologist. I didn't really know that much about the naiads. These guys swim with lateral undulation, which means they go like this, right? They move their bodies. Dragonfly naiads use jet propulsion. They shoot water through their bodies to get around, which is really kind of crazy. So as you could imagine, dragonflies are stronger. Their naiads are also stronger. They're better swimmers. So we were really curious to see which one would be better. And this is experiment. So we put brine shrimp eggs in the water, which are almost neutrally buoyant. Turn on the flow so we can measure. And then, whoopsie. Oh, stop. Shit. Okay. That's what the experiments are really like. We're like trying to increase the speed of the flow and catch it on video, but not have the little guy go down the drain. And not flood the lab.
Did he go down the drain?
Yeah, we didn't lose any. Yeah. This is a dragonfly. So,
Turn it on.
yeah.
Go ahead and go.
They're much fatter, see? Taking a little while to get the water in. Oh, okay.
Stop?
No, 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 keep going. You're now starting to
Pull
move.
up.
Oh. He seems fine.
Is that full blast in there?
Yeah.
See how long he can do this for.
Oh, okay. So what we've learned so far is that they have very different behaviors. So the dragonflies, when the flow starts to go, they'll put their heads down close to the substrate. Whereas these guys, they'll try to swim against it. So once they get caught by the flow, they're just like, so it's interesting. They have very different adaptations and strategies to the stream. This was the part of Mount Molokai that we visited, and we were lucky enough to hook up with a landowner who gave us permission to explore his property. And during that trip, we discovered an endangered species on his property. This is Megalagrion pacificum, which was thought to be extinct from Molokai. It's happily living right there on his stream. This is Gandharva, who led us, who took us on a hike.
All right.
Look at his land.
Oh, there he goes. You might want to follow him, Marguerite. Okay.
You have to be really healthy.
There you go. Hike you up the mountain.
Gandharva just goes without shoes and like 10 miles in a minute. So, yeah, I was like really sore after that day, but it was pretty amazing, and we saw just so many species on his property. And these are some of the members of my lab. So Julio and Elizabeth, Christina, and Kainoa. Okay, and I think that's all I have